Okay, so good morning and good evening, everyone. It's welcome to today's museum seminar. It's really my pleasure to welcome today's speaker, also my friend, Dr. Heping Xu from Westlake University in China. So a very, very brief introduction about Heping. So Heping currently is assistant professor in the lab of system immunology in Westlake University. Heping actually finished his undergraduate study in life science and entomology in Anhui Agricultural University, and then continued pursue his master's study in Zhejiang University. After which he moved to Beijing to start his PhD training in Institution for Immunology, School of Medicine, Tsinghua University. From 2014, Heping moved to Cincinnati Children's Hospital Medical Center, started his research fellow training. And then 2 years after 2016, he moved to Broad Institute of MIT and Harvard in Boston, US, continued his postdoc training until 2019. So from 2019, Heping actually moved back to China, as I just mentioned, started his independent research program as assistant professor in Westlake University. So the general research interesting of Heping's lab are in the area of immunobiology and system biology. The long-term research goal is to systemically decipher the fundamental machinery orchestrating immune reactions at both physiological and pathological states, which ultimately will contribute to our knowledge for understanding how disorders of immune system result in chronic inflammation and autoimmune diseases. So currently, Heping's lab is particularly interested in leveraging emergent system and computational biology approaches in immunology for decoding tissue-specific microenvironment in the molecular circuits that orchestrate two different aspects. One is the B cell development and immune responses. And then the second is the interaction between the nervous and immune system in the brain and the gut. So I think in a very short period of time, Heping really actually accomplished an astonishing amount of work, particular before and after he became independent research PI. He actually complete a really remarkable piece of work on the high-profile journals. And then in addition to that, Heping actually received also numerous awards and honors, which I'm going to list a few, which including 2015 Leukemia and Lymphoma Society of Fellowship in USA, 2015 Keystone Symposium Scholarship, and then 2019 he also received as Excellent Young Immunologist, Chinese Society for Immunology. I think we all learn more or less about Heping's work from the B cell, which also I believe is the topic of Heping's talk today, focused on B cell selection in both immune system and the nervous system. So it's really our pleasure to have you here, Heping. Please go ahead. Looking forward to your talk.
Okay, great. Thanks, Tran, for that very nice introduction. So I also want to thank Tran and Xiaolei for organizing this extremely successful online platform, which really helped us to communicate with colleges around the world. So it's really an honor and my pleasure to present some of our recent works in this seminar series. So as Tran introduced, the broader research interest of my lab in Westlake University is to understand the crosstalk between immune cells and the tissue-specific microenvironment. So very specifically, we study how the same type of immune cells adapted to different microenvironment signals to achieve tissue homeostasis, and how the dysregulation of this immune tissue microenvironment interaction leads to the development of chronic inflammatory diseases, including the allergic disease and autoimmune diseases. So in particular, we are very interested in the tissue-specific regulation of B cell response in this broader context. So for my talk today, I will try to cover two stories about the regulation of B cell responses in two different tissues. One is in the CNS border, and the other one is in the spleen. If I can move my slides. Somehow my slide. Okay. Can you see my slides at once? Okay, good. So I would like to start with a brief introduction about B cells. So just in case some of trainees who are not really working on B cells. So as many of you know, as one of the two major adaptive lymphocytes, B cells were initially discovered by Dr. Max Cooper in 1965 from the birth of Fabrius of a chicken. And he demonstrated that B cells generated from this very special organ is essential to mount extremely important protective human immunity. So later on in 1974, I think he found that B cells are also present in the mammalians like human and mice. Now, as introduced in all these classical immunology textbooks, we know that B cells in adult mammalians are solely developed from the common lymphoprogenitors, which are differentiated from the hematopoietic stem cells in the bone marrow. So as to B cell development trajectory in the bone marrow, so the most important task for B cell development is to diversify their B cell repertoire using only hundreds of antibody genes. So the exact number are actually vary a lot among different species. And through this so-called lineage recombination process. Now it's already extremely complex to illustrate here, but it's kind of oversimplified. So I just want to highlight several key events about B cell development. So it begins with a pre-pro B cell stage, followed by pro B cells. So in these two stages, the expression of recombinant activation genes, including RAG1 and RAG2, can introduce random VDJ recombination of B cell repertoire heavy chains. So after that, those B cells which developed into a stage called larger pre-B cells. So those cells actually turn off RAG expression. And they are trying to test if they acquired a functional Ig heavy chain by expressing a pseudo BCR receptors on the membrane to deliver antigen signaling. So the functional signaling will introduce cell proliferation. So then the cells will turn off all these cell cycle genes and exist mitosis. Instead, re-express RAG expression to support VG recombination of light chain in this so-called small pre-B cell stage. So after successful expression, both heavy chain and light chain, they become immature B cells, which later will enter into the circulation and leave bone marrow. So the VDG recombination really provides extremely diversified B cell repertoire, which are critical to protect the host from the variety of different pathogens. But this process also often creates autoreactive B cells, meaning they express a self-reactive B cell repertoire. So normally, the autoreactive B cells are eliminated or so-called inactivated through the autoantigen dependent negative selection process in the bone marrow. We call this central negative selection. The aim is to prevent the development of autoreactive disease later, such as lupus and multiple sclerosis. So when we really look at the literature and the textbook, so it's a lot like the T cell negative selection in the thymus, it have perfect L system. So actually, to us, how the microenvironment in the bone marrow that present autoantigens to a developing B cell repertoire is still largely unknown at this moment. So the question becomes more interesting if you think about those tissue-specific antigens, such as those many oligodendral glycoprotein. All these antigens supposed to be highly expressed, extremely restricted into the CNS and nervous systems. And the local concentration of those MOG or other things, potentially very highly tissue-specific CNS antigens in the bone marrow should be very low. So, so whether and how these CNS-specific antigens crosstalk with earlier development B cell development or the next central negative selection It's an interesting question to ask. So actually, Dr. Hermann Wegler in Max Planck Institute, together with his colleagues, have studied this question in 1998 by generating a MOG-specific Ig heavy chain knock-in mice. We call this TH mice. So they found that in comparison with the white-type mice, as we illustrate here, in both the spleen and the bone marrow, those TH mice actually have plenty of MOG-binding B cells in the spleen and the bone marrow. And the analysis show that both in vivo and in vitro, those B cells actually can respond to MOG. So it's not the inactive B cells. Those B cells can carry their function normally. So this actually immediately suggests that the MOG-specific B cells escaped the central tolerance selection in the bone marrow. However, even though these mice have so many mature MOG self-reactive B cells, they usually do not develop spontaneous EAE, which is an animal model of the multiple sclerosis in human. So there are multiple mechanisms and models has been proposed to explain this tolerance phenotype. For example, maybe this B cell response require additional help signaling from the T cells, which require to support the B cell response, and the T cells cannot escape the negative selection in the thymus, and etc., etc. So among all these models, there's one model I think is extremely interesting. It's so-called concept of immune privilege. So the brain has long believed to be an organ without an lymphatic drainage system for quite a long time. And also people believe that due to the presence of many barrier systems, like everybody knows the BBB system. So the brain seems to be unreachable by immune cells at the steady states. Therefore, it's insufficient or inadequate to mount an effective immune response in the brain by self-reactive immune cells, such as those large number of MOG-reactive B cells in TH mice. And this is somehow protect the mice from the developing of autoimmune diseases. In 2015, I think that everybody, potential immunology, knows this work now from the Jonas Kipnis group, as well as Carrie Alitrato groups from the publishing JEM. They actually rediscovered the lymphatic system at a steady state of the healthy mice in the dura mat. So dura is actually one layer of meninges directly attached to the skull bone. They use LIV1 labeling, which is showing the green here, nicely show that those mice at the steady state actually have not of lymphatic vessels. And more importantly, without any signs of disease, we found those T cells actually present in this within or nearby those lymphatics at a steady state. This actually bring really a new direction for the immunologists to explore, and people call this the meningeal immunity field. And since 2015, in the last couple of years, the composition, the function of meningeal immune cells has been extensively studied. Now we know that in addition to T cells, the many other immune cell types like dendritic cells, NK cells, even ILCs, and particularly ILC2s are present in the meninges. And another literature actually suggests those cells actually can modulate certain aspects of neuronal activities. So regarding to B cells, previous single cell profiling studies have shown that they are present in the meninges of mouse before our work. For example, in this very beautiful cell paper published by Ido Amity group, they actually captured the B cells in their single cell data sets, although the main focus of this work is about microglia. And also BCR are captured in other single cell studies using mass cytometry and other single cell studies. But so far, the contribution of these B cells to meningeal immunity and the immune homeostasis of the CNS is largely unknown. So in particular, those CNS autoreactive B cells, like in the TH mice, that escape the negative selection in the bone marrow could be potentially also present in the meninges, which are very close to the parenchyma. So whether and how these autoreactive B cells respond to CNS specific antigen in the meninges, a very interesting question for us to study. So to attack this problem question, Wang Yan, who is a PhD student in my group, and Xu Di is a member of the, my colleague Dan Yanhe's lab, they grouped together and decided to first look at the B cell phenotype in the meninges of mice at a steady state. At the beginning, very beginning, we realized that there's a potential contamination from the blood system. So they actually developed a method to IV inject, or adapt a method to IV inject APC country CD45 into a circulation of mice. And in a short period of time, we followed those mice were followed by perfusion and full tissue harvest. And by doing so, you can identify those there's a CD45 IV labeled cells. We think those are potentially contaminated cells from blood circulation even after perfusion. So those cells were excluded from downstream ancestors to make sure that the cells we profiled indeed are present in the meninges. And within those two potential resident cells, we identified B cells as showing a single cell data set labeled by CD19 and B220 double positive cells. And then using the whole mount immunolabeling and imaging, we further found that like other immune cell type, most of meningeal B cells, which are labeled with B220 here, the white dot, they actually aligned along with LYV1, which is those labeled red vessels, especially in the so-called CNS region. So this confirms that at the steady states in the healthy mice, indeed there's a B cells present in the dural matter of the mice. So next we decide to determine the developmental stage of those B cells by analyzing the membrane B cell receptors. As I just introduced, the earlier developing B cells do not express IgD and IgM on the membrane. And the immature B cells will be IgM single positive, mature naive B cells are IgD, IgM double positive. And after that, the antigen active or expressing B cells usually are IgD negative. And if they did not undergo class switch recombination, they will be IgM positive. Otherwise, they will be IgM negative instead IgG, IgE, or IgA positive cells. And to our very surprising, we found that the majority of meningeal B cells are IgD negative, and most of them are IgD, IgM double negative cells. So this actually mirrors those observations in the bone marrow, and in contrast to other organs like spleen, lymph node, and now in which in those tissues, essentially the double positive cells, which are mature naive B cells, dominate the population. Now in the small intestine, we see a small percentage, roughly 15% of cells are IgD, IgM double negative, and we know those are class switch IgA positive cells. So they already downregulate Ig and IgM due to the activation. So then the question is whether those cells are like earlier progenitors as we saw in the brain or in the bone marrow, sorry, or it's actually the class switch cells. And we rely on another marker, which is RAG. As I just introduced, RAG is essential for the earlier regulatory combination and do not express at antigen experience cells. And using a RAG report mice, we found that in both meninges and bone marrow, those IgD, IgM double negative, which are red here, or IgM single positive cells, which are blue here, highly express RAG compared to those mature naive cells. Suggests that those double negative cell B cells in the meninges are actually in the earlier development stage. And since the cells are really looks like phenotype similar to bone marrow, then the immediate question is whether those are like a contamination from bone marrow during the sample preparation. So to look at the overall distribution, relative distribution of those cells, we perform the tissue clearing for the school attached with dural together. So here you can see that this is a whole mount imaging of the mouse cranial bone. You can see here, we call this interperipheral area. If you zoom into this region, you can see a cluster of green signaling. Those are actually a cluster of development B cells, which are expressed in RAG. And then if you zoom into the parietal area, like the bottom is the SS region of the dura, you can see nice lymphatic vessels, and you see a lot of B cells actually locate inside this LYV1 positive vessels or nearby the regions. So this really suggests that they are definitely not the cells contaminated from bone marrow. It's actually located inside the dura and a direct attachment to a bone. Next question is to, since there are multiple development stages in the earlier development, for the earlier B cell development, so to further characterize the transcription states of B cells and the heterogeneity of the B cells in meninges, we performed a single cell RNA-seq, and the data was analyzed by Dian Yu, a computational PhD student in the lab. And you can see he identified roughly 7 different clusters. Those cells, including the pro B cells, which express, for example, VPB1, 3, and REG1 and REG2. As I introduced, this is for the light chain recombination. And after that, the cells downregulate REG expression and express a lot of mitotic genes for the larger pre-B cells to expansion. And after that, the cells will regain expression of REG and also to promote heavy chain recombination, and later on followed by the immature B cells and the mature B cells. We also identify the plasma cells in this dural method, and this has been reported by a very nice cell paper. And then we, using the flow cytometry, confirm the presence of pro B cell and pre B cells in the meninges, and the overall composition of this cell type is comparable between bone marrow and the meninges. So notably, we actually did, using both single cell analysis and flow cytometry analysis, we did not identify any earlier progenitors, such as pre-pro B cells and common lympho progenitors in the meninges. So this really suggests that meninges might serve as a distinct reservoir for B cell development only. It's not like for general earlier hematopoiesis. So at that moment, since the finding of development B cells are present in meninges, so quite unexpected, and so we would really want to explore whether such early development B cells also exist in the meninges of higher organisms such as non-human primates. You may ask why we just go ahead to analyze human. So the tricky thing is that the SS region is like an untouchable region for most of surgery because it will cause a real severe bleeding. So it's very hard to get the human samples at that moment. So we just go ahead and have several macaque and then perform the perfusion and harvest the macaque meninges for the flow cytometry and the single cell analysis. Since we do not have reliable antibodies to really analyze the B cell phenotype from macaque, so we've sought the CD45 positive, CD11B negative, and the CD3 double negative cells for single cell RNA-seq analysis to try to enrich B cell population in our data set. So overall, 9 clusters of B cell lineage cells were discovered by Dian Yu, which express distinctive markers as illustrated here. So you can see it's actually much more heterogeneous than meningeal B cells as we observed in the mice. Nonetheless, we are able to identify the pro B cells again using the same set of genes as we list in mice. Those cells express VPB, REG1, and REG2 for the light chain recombination, downreg to REG, upreg and cell cycle for larger pre-B cells, and re-express REG, downreg the cell cycle for light chain recombination, and also immature B cells, as well as several other B cell types. It's also very interesting phenotype to see those cells in the meninges. And importantly, using the same set of genes, we did now see the development B cells in the spleen as a control. So put all this data together, I think we demonstrate that the presence of a continuous trajectory of B cell development starting from the pro B cell stage in the meninges of both mice and non-human primates under homeostatic conditions. Now, based on this finding, there are multiple questions need to be addressed. So we actually picked up several questions, which we think is very interesting. So first one is that are meningeal development B cells self-renewable, or it's actually replenished from hematopoietic stem cells? And if it's required a replenishment, what's the road for this replenishment? So given our finding that the lacking of the CLP and the pre-pro B cell progenitors in the meninges, so we speculate that maybe those meningeal B cells, if it requires replenishment, the cells is maybe constantly require hematopoietic stem cell-derived progenitors, which should be in the bone marrow. So to test this, we generated a fetal mice model that express tamoxifen-induced CRE-ERT recombinase under control of a stem cell, acquired stem cell specific enhancer, which is SCL locus. And by doing so, the hematopoietic stem cells and their progeny will be generated genetically labeled with GLP upon tamoxifen administration. So as you can see here, without tamoxifen treatment, there was no GLP positive B cells in the meninges. In 2 weeks after tamoxifen treatment, we observed roughly 7 to 10% of meningeal B cells become GLP positive, suggesting there's actually replenishment from hematopoietic stem cells. And this frequency will further increase to 60% at 80 weeks post treatment. So actually, this is a maximal efficiency of this screen. So that's why we did not get 100% GLP B cells here. So this result really demonstrates that meningeal B cells will continuously replenish from the hematopoietic progenitors. So the similar B cell turnover dynamics were observed in the bone marrow and the spleen. In the bone marrow and the meninges, whereas the B cell replenishment in the spleen, lymph node, and the lung were much less efficient within 9 weeks after tamoxifen injection. And this is in accordance with the notion that the meninges and the B cells merely contain earlier developed B cells with a relatively faster turnover rate from the hematopoietic stem cells. Got it frozen again. Okay, so it's definitely required replenishment, and then we next to decide whether this replenishment of developed B cells in the meninges from hematopoietic stem cell is through the circulation. I think that's the most direct hypothesis, right? So, but to experimentally test this, we employed a parabiotic mouse model in which these 2 surgically joined mice can establish a very rich circulation, leading to a complete peripheral blood exchange. So at 80 weeks after surgery, we analyzed partner-derived B cells, which are CD45.1 cells in this CD45.2 mice. And we found that a roughly 50 to 50 exchange rate of B cells in the spleen, lung, lymph node, small intestine are reached at 80 weeks after surgery. But in contrast, that B cells in the meninges and the bone marrow from the same animal show significant lower exchange rate. That seems like that the replenishment of earlier developed B cells in the meninges surprisingly do not rely on the circulation cells from the bloodstream. Then if it's not goes through the blood circulation, which road that the progenitors used to replenish the meningeal B cell pool. So this question actually puzzled us for a while. Until we came across to this very elegant paper published by the Masses Lansdorf group. They found that actually in the meninges, it's actually directly connect with the skull marrow cavities as shown here, with so-called very specialized microvesicle channels, which serve as a shortcut route, allowing monocytes and the neutrophils to migrate towards the CNS. So in this regard, we know that there are plenty of B cell progenitors in the skull marrow cavities, as I showed you before. So maybe the hematopoietic stem cells from the skull marrow cavities may be a preferred origin of the meningeal B cells, and which might direct migration through this vesicle channel without going through the whole blood circulation system. So to test this hypothesis, basically we need to distinguish B cells generated from this cranial bone marrow versus a distal bone marrow like tibia. And to set up this system, one year actually come up with a quite smart design for the bone marrow transfer model. So he transferred the bone marrow cells of CD45.1 mice into a lethally irritated CD45.2 recipient to create a chimeras. But during the irradiation, he put a lid cap on the head of those recipient mice. The idea is to protect the hematopoietic stem cells in the skull bone marrows. And as a result, in comparison with bone marrow tibia, the transfer CD45.1 hematopoietic stem cells were not able to efficiently colonize the skull bone marrow, as the niche was still occupied by the endogenous CD45.2 cells. Thus, in such a chimera, the bone marrow of the school like manifest the distinct cellular composition from that in the TB. There's a significantly lower frequency of donor-derived CD45.1 cells in the developing B cell population than that in the school marrows, or than that in the tibial marrows. So importantly, the composition. Then we ask in the meninges, so which composition is similar to the meninges in terms of bone marrow and in the tibia and the school. And you can already see here the composition of developing B cells in the meninges were very close to that in the school, rather than the tibia, even at 3 to 6 months after bone marrow transplantation. The timing is important because the B cells in the meninges are also protected by this lead cap, right? So you may think that this is just due to the same effects. But if you think about what I just described before, the turnover rate for meningeal B cells is roughly 2 months, right? But we wait in the model up to half a year, it's still only similar to a school in comparison to tibia. So putting this together, it suggests that the school appear to be the preferred origin of the developing B cells in the meninges. But however, it remains to be determined whether those hematopoietic stem cell derived progenitor, I mean B cell progenitor, entering in the school, enter into the meninges, directly goes through the vascular channels that I described. Also, we do not have data to support that, but we think that's the case. Otherwise, there's no other pathways we can imagine right now. So, okay, to summarize this part, we demonstrate that hematopoietic stem cell derived progenitors from the cranial bone marrow, which actually replenish the meningeal developing B cells in a main bloodstream independent manner. And the next question we ask ourselves was that are these developing B cells in the meninges subject to negative selection if they recognize a single specific antigens? So back to this picture I showed you before from the specular group. You can see here, TH mice have plenty of MOG-reactive B cells in the spleen and the bone marrow. If our model is right, if they undergo negative selection, you should see a defect of meningeal B cells in TH mice compared to wild type. That's exactly what we see here. This is compared with a wild type or other BCR modified mice, for example, those MD4 mice, which is the BCR transgenic mice, which recognize hen egg lysosome, which obviously is not a non-self antigen, and also B18 high lock-in BCR lock-in mice, which recognize small molecule NP, again, it's not a self antigen. So early the B cells recognize MOG actually show a very significant reduction of total B cell number in the meninges. And if you specifically look at the BCR expression in those THB cells in the meninges, it's significantly reduced for the BCR expression cells compared with the THB cells in the bone marrow of the TB and school. So suggests that they are likely the IgM expression B cells were more significantly compromised specifically in the meninges of TH mice. So although we have all these types of controls, but it's still possible that this defect is not due to the BCR mediated effects. It's maybe due to some artifacts introduced when generating these mice. So to demonstrate this really BCR dependent process, we put a wild type mice and TH mice, the bone marrow of wild type mice and TH mice together and transfer into a recipient which were pre-irradiated and wait for 4 to 6 weeks and then look at the composition of different B cell development stage in different tissues. And you can see here in this IgD and IgM double negative population, meaning that those cells haven't expressed any BCR receptor yet, the frequency of CD45.2, which are TH B cells in the meninges, bone marrow of TB and school are comparable, meaning there's no specific reduction of those THB cells at this stage. However, when the cells come into the IgM single positive or double positive cells, you see a very significant reduction of those Th-derived B cells, very specific in the meninges, but not in the bone marrow of tibia and skull. This is observation really reveals a very BCR-dependent and tissue-specific deletion of these MOG-binding B cells. And also, to further demonstrate that the reduction of autoreactive B cells in the meninges is seen as autoantigen-dependent process, we actually attempted to rescue the defect of B cells in TH mice by deleting MOG expression. As you can see here, in wild-type TH mice, again, there's a reduction of IgM expression B cells, and very specific in the meninges, but not in the bone marrow. However, when you knock out MOG from the TH mice, it's completely restored IgM expression in these TH mice meninges. Therefore, this result clearly demonstrates that the deletion of self-reactive B cells in the meninges is seen as autoantigen dependent process. Okay, so to summarize this part, we actually found that earlier development B cells present in the meninges of the mice and non-human primates at the steady states. These cells are continuously replenished by the hematopoietic stem cell derived progenitors in the skull and probably go through these directly connected vesicle channels. And these earlier development B cells are subject to negative selection in the meninges that are mediated by the CNS specific autoantigens. So I think our work really demonstrates that the meninges could function as a distinct reservoir for the B cell development and allowing this, we call it local or in situ negative selection of CNS autoantigen B cells, and which could serve as additional layer of the checkpoint to stringently maintain a local self-reactive immune repertoire. And I think this, obviously, this in situ negative selection, we believe is very important for the understanding of the development of autoimmune disease like the multiple sclerosis. And we have some unpublished data to show that these B cells play a very important role to promote development of the multiple sclerosis models. And hopefully we can show this next time. So I think I still have a couple minutes. So I will jump into a second story. As I introduced at the beginning, those cells which successfully pass through the central tolerance cell selection in the bone marrow will enter the circulation and then migrate to the spleen for further development. So in the spleen, those called immature B cells are still need to undergo the peripheral negative selection, which I did not illustrate here. After that, the B cell will become into a stage named as mature naive B cells, meaning those cells express membrane IgD and IgM, and they are ready to respond to antigens like pathogens and vaccines. And very importantly, a subset of antigen-active B cells will differentiate into a stage named as germinal center B cells, or called GC B cells. So germinal center B cells undergo very rapid proliferation and expression. AI activation induced cytidine deaminase, or AID in short, which induces somatic hypermutation of the antibody sequence that will change the binding affinity of BCR to the antigen. And we have a system called positive selection in the germinal center. It's extremely unique and important for human immunity, as this is potentially the early stage or step that our immune system can positively select for the best B cells. So the best meaning those cells acquire the highest binding affinity of BCR to antigens. So this is how germinal center look like in tissue. I think this is in spleen. So physically, they have two adjacent zones or compartments named as the light zone and dark zone. I will not go into the details of the selection model, but basically, in dark zone cells proliferate and introduce a mutation. They migrate to the light zone to compete for the survival signaling, and only those cells acquired high affinity mutations will survive and the migration back with aim to gain even higher mutation. And after migration back and forth, and somehow we're not percentage sure yet, the sum of cells will differentiate into, think they are good enough to differentiate into the high affinity plasma cells, which can secreting the high efficacy of the neutralizing antibody. So in this context, the question we are interested to ask, so based on the current knowledge, could we target the germ center selection or affinity maturation, for example, using some chemical approach to facilitate production of high affinity neutralizing antibody? For example, in the context of a vaccination or during the infection, I think if you can have more good antibody coming out earlier, it will be very beneficial. So unfortunately, the answer at this moment is no. So far, there's no purchase has been reported that we can apply to facilitate the antibody affinity maturation in vivo to the best of my knowledge. And the reason I think is that the model that I just described, like a cell-cell interaction, cell migration, are all at a cellular level. The real cell intrinsic molecular programs that are utilized by high affinity germ center B cells to support their selection is still unknown. I think there are multiple challenges to explore the underlying molecular selection in this context. And we think the most important one is that how could we identify and purify germ center B cells that have acquired a high affinity mutation during a polyclonal immune response for the downstream analysis, right? If we can do so, we could apply different means of experimental approaches to understand the unique feature of these high affinity germ center B cells relative to their low affinity ones. And those features could help us to reveal critical molecular pathways as drug targets. So to tackle this problem, the computation student I just introduced, the Dianyu group together with Wang Yan, they actually come up with the idea that they think the fact that the current single cell RNA-seq is to able to capture transcriptome and Ig sequence information simultaneously. In theory, we actually can computationally map out all the mutation profiles in the antibody sequence for individual B cells, and then ask which kind of unique molecular program are displayed in those B cells which that acquired high affinity mutation relative to other cells, which are considered as background. So to achieve this, obviously, the key is to know which mutation is high affinity mutation, right? So to do so, we introduce a very classical hepten NP immunization model. NP is a small molecule. They can conjugate to different proteins like KOH or OVA to induce a robust human immune response against this hepten. And through many years of pioneer studies in the field, we know that in the B6 mice, after immunization with NP, BCR clones which carry this particular variable gene, which is 1-72, actually dominate the GC response to this particular hepten. And more importantly, we know that there is some substitution at position 23 from tryptophan to leucine will substantially increase the binding affinity of this gene to NP by tenfold. So that means we actually can use this mutation as a readout to distinguish high versus low affinity cells. And then Wang go ahead to immunize mice and perform a single cell experiment, and then you analyze data. So as people do, we do a NAT-U map. As if we not go into the details about these clusters. Basically, we capture all the known feature of the germ center B cells. And for the BCR repertoire, as expected, this particular variable gene, IGHV1-72, dominant response to NP-specific germ center B cells. And if you map out all the mutations, we see this very dramatic enrichment of this mutation at the position 33, and the germinal sequences, tryptophan, and the dominant mutation is leucine. So everything matched to the previous, as we expected, matched to the previous knowledge about the NP system. And all this computational analysis demonstrates that we are able to reliably analyze this somatic hypermutation from the NGS list. You might think there's a lot of noise there. And then we go ahead to ask which kind of molecular program are significantly enriched in this W33 mutation, or other two other mutation also has been shown to increase the binding affinity and the group together as a high affinity mutation clones and relative to those we call the background. So for example, we noticed that the activity of my total program was significantly enriched in the high affinity clones. So this was consistent with the finding from the Michelle Lucent group, suggests that the high affinity clone B cells will manifest accelerated cell cycle and while undergoing the path of selection. And another significant pathway that we are interested to look at is oxidative phosphorylation. we are interested in this pathway because germ center B cells in this Nature paper has been reported to be preferentially relying on glycolysis because the germ center area appears to represent a hypoxia environment. But in contrast, the other beautiful work from the Mark Slumchak group found that germ center B cells in vitro at least minimally utilize glycolysis, and instead they use oxidation fatty acids. Therefore, we think the metabolic program that meets those energetic demands for this very rapid proliferate germ center B cells are still need to be explored. And furthermore, no matter glycolysis and oxidative phosphorylation, whether those pathways directly involved in the promoting affinity maturation is still unclear. So we actually found that the activity of OXPHOS will positively correlate with affinity maturation in these NP-specific germ center B cell responses. So before we go into the next step, we want to make sure that this pathway also upregulates in other immunization antigens. So to demonstrate it is potentially widely true, we actually use OVA, another antigen, to immunize mice and perform a coupled single cell analysis for germ center B cells post OVA immunization. So now in this immunization context, we actually do not know which mutation in which variable region, not like W33L in VH186.2. In this context, we don't know this information. That potentially will increase the binding affinity. But we reason that due to the positive selection of high affinity mutations, the dominant mutation, meaning the enriched mutation across all Ig variable genes in the data set are likely to be the result of the selection that's roughly equal to the high affinity mutations, right? Because of the selection for the high affinity mutations. So therefore, then we actually develop a pipeline to align the amino acid sequence of the individual, which are dot here, variable genes, to the corresponding germinal sequence, and then review the several positions that acquired substantially more mutations with high count of numbers. So for example, this position 59 here in this IGHV1-26 variable genes is significantly enriched. And furthermore, the germinal sequence here is serine, I think, and the dominant mutation to serine suggests there is a selection for serine. If it's not a selection, this amino acid will be random, right? So we think this is a positive selection. And we have two other evidence. One is the frequency of the mutation increase over time, suggests again, during the time, the mutation will come out. Also, we have quite, also it's indirect, but strong evidence to suggest this substitution increase the binding affinity of this gene to OVA. And more important, if you look at the activity of oxidative phosphorylation, is also increased in a group of, at least enriched in this, in the clones carrying these mutations. So suggests that it's without strength and link between increased OXPHOS activity and the positive selection in germ center B cell clones. So next question is, if we perturb the OXPHOS activity in germ center B cells, could we impair the affinity maturation? And this is what collaborated with Harinder Singh and his great postdoc, Gurdev. They have COX-10 flux mice, and we have data to show that this is actually important component of terminal 4 of the electron transfer chain. If you knock out these genes in A20 or other B cell lines, it will significantly dampen the OCAR in the seahorse acid. And then Gurdev knock out this gene using AID crew, and you find that the germ center B cell frequency would decrease. It's not a surprise because these cells indeed require a lot of energy. And but interesting, if you look at the enrichment of these high affinity clones, it's significantly inhibited by this knockout of COX-10. And more important, the serum binding affinity is also lower in this condition knockout mice. Therefore, this result actually demonstrates that the GC B cells require OXPHOS activity, not only for their expansion, but more importantly for the efficient positive selection. And back to the question that I proposed at the beginning, could we promote affinity maturation by somehow promote these metabolic pathways? So we tested several compounds that has been shown potentially can promote oxidation phosphorylation or mitochondrial activity in vivo. And turns out there's a compound called autopris, which are here, has been reported to increase mitochondrial OXPHOS activity while activating PCG1 alpha and the NF2 pathway. And then after immunization, if we give mice this autopris, first thing we notice is that not significantly increase the proportion of germ center B cells. But however, it significantly increase the frequency of these high affinity clones carrying this mutation. And more importantly, autopris significantly promoted the affinity maturation of these NP-specific antibodies at a different point post-immunization. I think the obvious question is that we haven't demonstrated that this drug really go into the BCL intrinsic molecular mechanism. And that's also the question the review asked. And we are still trying to demonstrate this later through the other approaches. So with the data I did not show you here, we think by generating a quite a lower analytical computational pipeline for coupled analysis of somatic hyperpigmentation with transcription states, our work demonstrated that GCB cells acquiring the high affinity mutations displayed enhancement of OXPHOS activity. I think the key is also the deficiency of course, OXPHOS activity reduces cell division and impair pathway selection. And using this drug can significantly improve the affinity, promote affinity maturation. Also, the side effects right now is not ideal. And these two actually really suggest OXPHOS activity is important for germ center selection. So, and also the pipeline obviously can be applied to the clinical studies to understand the mutations with different diseases. And we are currently working on look at the BCL mutations in the context of the IBD diseases. Okay, so with that, I would like to thank all my lab members. I think I already cited all the key people in the project, and it's really extremely lucky to have opportunities to work with these very talented young trainees in the lab. The first project was equally very closely collaborated with my colleague Dan Yang, and the second project is collaborated with Harinder University of Pittsburgh. And we received lots of critical tools like TH mice, Hermet generated mice in 1998. And when I started my lab in 2019, I wrote a letter to him and he immediately agreed to share the mice to me. I think that's really promoted progress of this project. We also have other mice from the region from the colleges around the world, and this is funding agency. And I have two more important slides to show. One is that we have all the positions open for postdocs, PhD student, and research assistant. If you are interested in our research, please feel free to contact me. And especially for PhD students, the School of Life Science PhD student program right now is open for application. And sorry about this Chinese if you are a foreign student, but we do have a program open for international students as well. If you scan this code, you will know more information about our program. So please submit your applications. So with that, I will end here and thank you. And I'm happy to take any questions.